online seminar. However, I believe that the excitement to participate in ISSMART 2020 will not be lessened. This year's theme is Smart Molecules from Natural Resources for Global Pandemic Diseases, which covers broad range of research field with various scopes of biochemistry and molecular biology. Let me get my math. Molecular sciences, biomedical engineering, neurosciences, and nutrigenomics and nutrigenetics. These particular themes is intended to promote recent advances in the field of natural genetic resources. Importantly, to address important issue that is outbreak of the coronavirus. This international forum also provides a platform where national and international academia and researchers, policymakers, and other stakeholders to translate technology, exchange ideas, and help stimulate multidisciplinary international collaboration in a convergent manner for shaping a worldwide sustainable development. The selected full papers from the conference will be published in Scopus Index Proceeding in Journal of Physics IOP Conference Series. Last year, we already published selected papers from ISSMART 2019 in Journal of Physics IOP Conference Series Volume 1374. For this year, around 70 people registered to the seminar. This consists of 45 presenting participants, about 30 non-presenting participants, and 5 keynote speakers. I would also like to thank to our keynote speakers. We have Professor James Keto Diet from SUT, Suranare University of Technology, Thailand. Professor Fahrul Zaman Huyop from University Teknologi Malaysia, Malaysia. Professor Dian Handayani from Universitas Brawijaya, Indonesia. Professor Fatkia from Universitas Brawijaya, Indonesia. These two ladies are from our research center, Monagans, and also to Dr. Saki Sirako from the Sumeikan University, Japan. ISSMART 2020 could not become a reality without the help and assistance of many parties. Thus, in this occasion, I would like to sincerely thank the Director of Universitas Brawijaya, Research Center Monagans, and all members of the organizing committee who have provided meaningful help and assistance for the implementation of this conference. And last but not least, I would also like to thank to all of the conference participants who will contribute to making this ISSMART 2020 the most memorable event of ISSMART. Last but not least, again, we have tried to do our best to prepare ISSMART 2019. However, there is nothing completely perfect in the world, including this conference. Therefore, please accept our deep apologizes for any inconveniences found in this conference. Finally, please enjoy ISSMART 2020. Thank you all to your attention and thank you all to your participation. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for Mrs. Anna Savitri for the welcoming speech. It is now my deep pleasure to introduce the fourth Vice Rector of Brawijaya University, the Excellency Professor Dr. Insinyur Sasmito Jati, MS, to deliver speech, and afterward we would like to request the Vice Rector Brawijaya University to officially open the second International Seminar on Smart Molecule of Natural Resources 2020. For Professor Dr. Insinyur Sasmito Jati, MS, the time is yours.
March 2020. I would like to extend my warmest welcome to all delegates of ISSMART 2020, our second international seminar on smart molecule of natural resources. Welcome to Malang, one of educational cities in Indonesia. There are about 40 universities in Malang. The largest university in Malang is Brawijaya University, and also the top best 10 in Indonesia. ISSMART is held annually, starting last year with ISSMART 2019. This conference is organized by Research Center of Smart Molecule of Natural Genetic Resources, or SMONAGENIS. SMONAGENIS is one of the research center in Brawijaya University. SMONAGENIS is also one of the leading research center in Brawijaya University, also one of top 5 research center in Brawijaya University. ISSMART 2020 aims to promote mutual exchange between scientists and experts to discuss a new research result in the field of theoretical and experimental of smart molecule from natural resources. The seminar facilities of researchers and academic members or experts from university, government research institutions, private sector, and non-government organizations to share their knowledge and experience through the discussion on plenarization and parallelization of oral presentation. I believe that ISSMART 2020 should bring advance for all participants. They will learn many new aspects of research in related topics, either from keynote speaker or general participant. In addition, collaboration between Universitas Brawijaya and other universities, both from Indonesia and overseas, can be initiated. Due to the coronavirus outbreak or COVID-19 pandemic, ISSMART 2020 is going to be held as virtual conference. However, I believe that this online seminar will still attract interest and will not reduce the excitement of this year event. Finally, I would like to express my gratitude to expert keynote speaker for their expertise and knowledge they will bring to the conference. And of course, the inevitable discussion their talks will spur. Special thanks are also extended to the member of organizing committee for their hard work in bringing this conference together. And last but not least, I would like to thank all of conference participants who will contribute to making this memorable ISSMART yet. Please enjoy ISSMART 2020 and have a delightful seminar. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you. Ladies and gentlemen, now we come to our main program, plenary session. We will be led by our moderator, Dr. Nia Kurnianingsih, MDOMed. For Dr. Nia Kurnianingsih, MDOMed, the time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good morning, ladies and gentlemen. Please welcome in our first day plenary session. Me as moderator, today, Nia Kurnianingsih. We have two great keynote speakers today. First keynote speaker is Professor Dr. Fahrul Zaman Huyo from University Technology in Malaysia. And the second keynote speaker is Professor Dian Handayani, PhD from Department of Nutrition, Faculty of Medicine, Universitas Brawijaya. For each presenter, we have 35 minutes for delivering the presentation, followed with 10-minute discussion. I would remind you all that all participants are only allowed to ask a question via chat room. And then I will deliver the question to the keynote speaker. Okay. We will start with our first session. Professor Dr. Fazrul Namasyah. This is the curriculum vitae from Dr. Professor Fazrul. Yes. He is a professor of microbiology, Department of Bioscience, University Technology Malaysia. He is a senior member of University Research Group under Research Alliance of Health and Wellness. He is also a lecturer in Department of Bioscience, University Technology Malaysia, Johor Bahru, Malaysia, with a lot of great publications. For Professor Fazrul, time is yours. Thank you.
Can I start now? Testing, 1, 2, 3.
Okay. Okay.
Belum di full kan? Prof. Fazrul, you can start your presentation right now.
Yeah. Can you listen to my
You can
voice?
unmute your audio. We cannot hear you.
Did you hear my voice?
Still cannot hear you. We already, okay, sorry.
Testing, 1, 2, 3. Can you hear?
Yes, we can hear.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullah. So first of all, I would like to say thank you very much, especially to the chair of ISSMART 2020, Dr. Anna Safitri, for inviting me this year session. And I would like to congratulate to all this committee for successfully making this event alive today, as you can see. And of course, today as a first speaker, I'm not solving any pandemic issues at the moment, but I'm sharing my interest during my 20 years of investigation as a lecturer in this department. So my interest basically to carry out something like bioremediation. So this time we have opportunity to study bacteria population in Lake Tuz, Turkey. So in general, as you can see, in some part of the country, in the world, for example, in certain places, for example, you have wastewater treatment, and then if there is intrusion by saline in the wastewater treatment, therefore the wastewater treatment will not become very efficient. All right. So therefore, I think this investigation is relevant at the moment due to the fact that some bacteria, especially from the sea, they have some ability to degrade chemicals, especially pollution, in the presence of salts. So in the world, there are many lakes. It has varieties concentration of salts. So we choose Lake Toos because Lake Toos is like, it has high concentration of salts, which is more than 50%. But seawater is low, less than 3%. However, in Lake Toos, it has very high concentration of salts. So therefore, we wanted to investigate certain microbes that are present that has the ability to withstand in high salt, at the same time can carry out the job of pollutant degradation. So Lake Toos is located in the middle of Turkey, very far actually from Istanbul. So you can see Istanbul is the capital of Turkey. And you can see the Lake Toos is the level from the sea level, around about 950 meters from the sea level. And the area is very vast, a very huge area. And then this area at Lake Toos, it has some economical importance because it provides salts to the country, right? So the area covered 164,200 hectares. And then the most salty lake, 32.4% of the salt concentration. And of course, we talk about bioremediation, meaning that we wanted to utilize this bacteria to degrade or to remove these hazardous materials. So therefore, this investigation was carried out. So when we talk about bioremediation, what does that mean to everybody? So first of all, we must understand that the first criteria, the bacteria must possess a gene, a special gene that can cause degradation. So for example, in this bacteria, it has some gene specific for microbial degradation. And number two criteria, all the chemicals surrounding the bacteria must be able to be transported into the cells. So that means you can see at the layer of the bacteria must have some mechanism to allow the uptake of the chemicals into the cells. So once the chemicals enter the cells, so what happens next? The chemicals will trigger this gene so that when it triggers by means of transcription and translations, so the proteins will be produced in the cells. So now we talk about the proteins produced intracellular, not extracellular. So after the entry of these chemicals, for example, halogenated compound, so these proteins will then react with these chemicals. We call it zymatic activity for degradation. And then what happened next is these chemicals transformed into something else. So this transformation process normally will allow bacteria to utilize these chemicals as their sole source of carbon. So that's the ideal, the requirements for degradation to happen. So the current issues, basically in our country, in Malaysia, for example, recently in 2019, there's a huge havoc in the country due to contamination of the Kim River. There's a river in Johor, so it's like all these factories, they throw things, especially toxic chemicals into the river system. So we call it illegal dumping. And then what happened next, because sunshine started to react with the river water and started to produce some fumes. So what happened is that the people surrounding the area, because of this, they inhale all these toxic fumes, and then it has caused some disaster to the schools and housing areas. So therefore, these are all problems. So of course, when you say bioremediation, always disadvantage. For example, sometimes the remediation cannot take in the very fast mode. It will take sometimes 3 days, or maybe as long as 6 months or 12 months, for example. So not only in Malaysia, but in other countries, in Manila, for example, a problem of solid waste people throwing plastic. So microplastic is another big issues in the world, for example, that can cause toxic to the sea and can cause mammals, fishes, and whatever microorganism in the sea will die, for example. So these are all the scenarios you can see in the world currently is a problem. But nowadays we try to stop. We try to educate people not to use plastic. Even we go to restaurant, you don't have straw to drinks. You straight away drinks from the cup rather than you use straw, because to avoid using plastic. At the university level, they stop using plastic container for food so that people cannot throw all this substance into the sea. So talk about dehalogenase. It's always, we must understand the halogens. Halogens is group 7, like bromine, chlorine, iodine. So these halogens, when in form of complex, halogen chemicals contain oxygen, nitrogen atoms, and so on. So this become toxic. So the idea is that all these synthetic man-made like herbicide, fungicide, it has this halogenated group compound. So what happened? What the enzyme now is like to cleave halogen and carbon, to cleave or to cut, to break off and transform into another product. So this product will be used as a carbon source for the bacteria to grow. So this the ideas. So now when we look at the what you call the lactose, is actually pristine. I mean, lactose is, I can say that it is free from pollutions, because the water level is very little. It's 0.5 meters depth. So the area is dry, especially in summer, and then there are no rivers. There's no river to enter the lake. So therefore, I can see that the lactose is pristine. So how does this dehalogenase will be produced? Because there's no chemicals. So what we believe that there is always a natural halogenated products. They are biosynthesized by marine organism, algae, and corals, and so on. So this will allow the bacteria, because thousands of years, the bacteria already interact with this natural HNPs. So therefore, the bacteria can produce dehalogenase enzyme. So that's the hypothesis that we derive from this kind of why dehalogenase is present in the bacteria from lactose. Okay, so then we look at the basic enzyme mechanisms. So normally, all these chemicals, it has the position of halogenated compound, halogens can be L or D forms. They are stereoisomers. So for instance, here we look at L, if it is L position. So X represent halides, chlorides or iodine, whatever. And then it is 2 carbon, CO minus plus OH, because because we call it hydrolytic enzyme in the presence of water. So the special about this enzyme, it can invert the products from L become D, and then remove the X, replace OH. So OH group hydroxyl came from water, and then finally you release these halides. So you have another forms of compound, we call it dihydroxypropionic. So this one, like lactate, this kind of chemicals, it's actually a carbon source, lactate, pyruvate from 2,2-dichloro, removal of 2 halides, is all the carbon source for bacteria. So this basic mechanism for the enzyme. Like monochromic acetate, DL2-chloro, and 2,2. This is a model from this study. So herbicide, we take example 2,2-dichloro. It has 2 chlorides at alpha position. So what happened next? You have the halogenase that can cleave carbon halogen. So first step and second step. First step, remove first halides, and second step, remove second halides. And what happened next? You have another product. We call it pyruvate. So this pyruvate is actually carbon source for the bacteria to grow. So again, our interest, we wanted to look at this extreme environments because extreme enzyme can react in extreme environment is something like novel to us. So that's why we always think about this, whether it is pH, temperature, salinity, pressure, and so on. So these are the properties of the extreme. So here we choose saline, salinity. So bacteria can be categorized whether it is non-halophile, can grow less than 1.2% salts. So in our case, we call it halotolerant because bacteria can grow at high salt concentration. And sometimes if you reduce the salt, the bacteria also can grow. So we call it halotolerance. And the rest, halophile and extreme halophile, we not managed to identify this kind of bacteria at the moment. So the extreme environment is actually a habitat considered as very hard to survive. But this bacteria can survive in this extreme environment. So this is something special about this bacteria. So dehalogenase from extreme environment is very rare, not many. So the first bacteria must survive, and secondly, they have another properties. For instance, to degrade pollutants. So that's the idea. So in the world, in terms of hypersaline environment, there are some microbes that has been isolated, and maybe they have different properties. So in our case, we focus in pollutant degradations. So this is the background of the water system, briny water, saline, brackish, and fresh water. So the fresh water normally no salts. There is a salt, but very, very small amount of salts. So when we talk about the fresh water, it's less than 0.05%, and saline is between 3.5%, and high salinity lactose is more than 15% salt concentration. So the scope is actually to look at how the bacteria can tolerate. At the same time, we wanted to measure how this bacteria behaves. For instance, it can produce special enzyme, we call it dehalogenase, that can degrade a model compound due to dichloro just now. And then we wanted to study what are the properties of the enzymes. So we talk about the moderate, the halophiles, for example. And then the halophiles organism thrive in high salt concentration. Extremophiles, we call it archaea. Most halophile belongs to Halomodaceae or Proteobacteria. So this can be found in Dead Sea or lactose, for example. And then what makes the bacteria can survive? So this is very interesting facts. So basically, once the cells in the high salinity, the bacteria become shrink because the water will be moved outside the cells. I'm not going to talk about very details, but in this particular situation, so the bacteria can survive because the bacteria can allow certain ions, like potassium chloride, for example, to enter the cells. So it will balance the cells so that the cells not shrink, so they can adapt. So they must have a special gene to allow this to happen. So this is very interesting facts why this bacteria can survive in high salts. So the bacteria will not die in this particular concentration. So the idea is to analyze the bacteria itself and of course the properties of dehalogenase. So since this is very general perspective, I don't want to go into very details so that even people from plant biotech can still understand the scope of my talk this morning. So how we designed the experiment? We started from the screening of the bacteria. So we must isolate the water sample and bring to the lab and started to grow them in a plate culture medium in the presence of salts. Plus also we provide the carbon source. The carbon source we use 2,2-dichloropropylene acid and DL2-chloropropylene acid as a model substance to allow growth. Of course, because too much pressure to the cells, we also provide small amounts of something like nitrogenous and nitrogen chemicals to allow growth, to support growth. So then after we saw some growth So we identify the bacteria based on biochemical characteristic, and also we studied CCS RNA bacteria. We sequence the CCS gene to know the name of the genus species. At the same time, we wanted to optimize their growth, their growth conditions in the presence of different salt concentration, pH, and so on. Then we analyze the HPLC, the HPLC to check whether degradation happened or not. So we check the concentration of 2,2-dichloro, maybe we provide from 20 millimolar, and after complete degradation or mineralization, we saw not much 2,2-dichloro left. And then we fish out the gene. We use special primers to fish out, to do PCR, to allow the complete gene sequence. So we must use special primers to amplify the halogenase gene sequence. And then we study the proteomic in terms of the properties, the structure and functions of the proteins is based on bioinformatic analysis. And then we do the structural functional analysis. We do the modeling, and then we do the model refinement, validation, and tunnel to see how these chemicals or 2,2-dichloro being transported into these specific catalytic sites. And then we do the docking analysis, and finally the electrostatic potential. So these are all the general scope of the investigation take place. So after 9 days incubation, we can see that some bacteria started to grow. It has white, it has not white, this time it has reddish spots on the plates. So it is actually rod in shape bacteria. Then when we have on the plates culture at 25% sodium chloride, it seems to be red in color. It has produced some color. So even when we grow them in the liquid medium, we can see very intense red color. It produces pigments, and of course the bacteria cannot grow up to very high concentration. The limit is 25% of salts that we use. So these are the general properties. I don't want to discuss too much about these technical informations. So the bacteria can survive basically up to 25% sodium chlorides at various temperature up to 35 Celsius and the pH 9. So most of bacteria that we isolate, the halogenase properties, the enzyme can work well up to pH 8.5 or 9. So that's not the problem of that. So then we test individually in terms of how the bacteria can survive at various concentration of 2,2-dichloropropanonic acid. So basically, the bacteria can grow well at 20 millimolar 2,2-dichloro. But if you provide too much 2,2-dichloro as carbon source, like 50 millimolar, the cells cannot survive because it is too toxic. And then, of course, the pH optimum, we up to pH number 9. The temperature optimum, 30 Celsius, can survive at 35 Celsius, and bacterial growth can we can measure the growth based on the stability of the cells in a specific 2 to 20 millimolar carbon source. Then we do the identical typical analysis to check whether what are the name, genus, and species, because it is important for further investigation like genomic analysis. In genomic analysis to profile the whole genome of the bacteria. So this information is very crucial to really identify the genus and species name, so that we use this 60S plus we must compare that to this, what you call it, the biochemical test to ascertain the name of the genus and species. We call it Pseudomonas halophile strain HX. Oops. Then we checked whether 2,2-dichloro has been utilized using HPLC analysis, and then within 96 days, 0 to 96 days, around about nearly the complete degradation, up to 96 hours, the complete degradation happened. All the 2,2-dichloro. So we have some confidence level that 2,2-dichloro utilized by this bacteria as a sole source of carbon. So that's the most important. So we plot the growth curve experiment against concentration an hour, and we measure the growth, the stability of the cells, because again, it is very crucial at this stage to see the bacteria really can grow in the soil in this particular carbon source. Therefore, it is worth to do the next experiment to fish out the gene using a specific primer system. So in PCR analysis, so we are using primers that has been published by Hill 1999 to fish out the partial gene sequence. So it is amplified, and then based on this partial gene sequence, we compare to the presence with other dehydrogenase gene. And then from this, we can see that some areas or some nucleic acid nucleotides, some nucleic acid, they are is suitable to use as a primer. So we design another primer in order to get a complete gene sequence. So based on this, it has high identity to a DHI, same group of what we are isolated in the DHX partial sequence. So based on this, so then we can have some confidence that we have the correct genes based on this sequence comparisons. Then we design a new primer, was designed in order to get the full gene sequence. This primer, of course, we have to design it from the start, from the two ends, 5' and 3' end. Then we use genomic DNA as templates to amplify the full gene sequence. So this sequence has been published. So we managed to identify the specific nucleic acid sequence, and then from this we generate what do you call it? The specific acid amino from this nucleic acid. So we determine we pair up the pairwise alignment between our DHX, and of course DHI is a reference acid amino sequence, and we can see that a lot of identities between DHX, the one that we isolate, and DHI protein. These are dehydrogenase gene, dehydrogenase protein acid amino sequence. Then we analyze the composition of DHX acid amino, what compared to the most average members of dehydrogenase. We can see that it has a lot of, in the surface area, the surface protein has a lot of charged acid amino. So what we do with this protein acid amino sequence, we further analyze to determine the protein sequence. We get the primary sequence of the acid amino composition, and then we do the homology modeling analysis, and then we do the refinement, the molecular dynamic simulation, DHX in the water system, so that it will mimics the actual conditions of the proteins. Then we do the assessment of the proteins in the structural visualization experiment, and then we check the electrostatic, and then finally we do the docking, and you can see that how this special carbon source will attach to the catalytic site of the proteins. So I'm not going to go into too technicals. So these are all the closely related, because since it can react with hydrolytic compound, so some of these acid amino, they are identical to the published acid amino sequence of other bacteria from Pseudomonas Bocalderia, Pseudomonas species from this particular acid number, and then other bacteria that you can see on the slides. And then as to compare from us, so you can see most of so far the published mesophiles, thermophiles, cycrophiles, and then we have our study related to hydrophile because it can survive at 25% salts. So the general findings, you can see that it is the report detailing dehydrogenase halotolerance bacteria, and then can grow at 2,2-dichloropropionic acid at 20 millimolar, and then it seems that can survive in this carbon source. So I think that's all. My talk, I think nearly 30 minutes to complete, so already described. So I would like to, the future work, we wanted to see to do the genomic analysis. Now underway, we analyze the full gene sequence because we wanted to see how the gene expression, because we need to have a complete operons. That's what interests us. What trigger this dehydrogenase being produced? It must have the regulatory gene, and then we must have special promoter system, and then we need to check the structural genes. So what are the genes involved in order to allow the transport of these chemicals into the cells? We call it permease system, and how many regulatory proteins really control, and how many dehydrogenases are present in this bacteria. So there are a lot of information can be derived from this genomic analysis. And then the metagenomic profiling of Tuzgulu lake has been done. And then, I mean, the information has been submitted for publications related to metagenomic profiling. And then currently we are looking at Soda Lake, Lake Van. So this genomic analysis is still in the process. So I would like to thank Dr. Yilmaz Kaya. So Dr. Yilmaz Kaya is the project leader of the current of this study, because the grants came from the Turkish government, and then of course we have the researcher Dr. Edbaib from Bin Walid University in Libya. So he went to Turkey to do some of his analysis, and in part analysis was done in Malaysia. And then Dr. Aliu Adamu from Africa, from Nigeria. So he's the one doing the modeling analysis. And then Roswanira Abdul Wahab is the collaborator, PhD supervisor. Dr. Azmir, of course, is the one to help us in doing the docking, because docking, after we establish the structure, we need to dock, things like that. So these are all, we need many people to help us. And then Habibat is our PhD student. And then Batul is the one doing Soda Lake work. So this is involved a lot of investigators from different country. And of course, the grant came from Turkish government, Tubitak. So I think that's all I wanted to share here for today. So again, I would like to say thank you very much, especially to Brawijaya Group, ISSMART, the chairperson who invited me to deliver my talk, and all colleagues in Brawijaya. So I sincerely thank you very much for this sharing session. So I pass back this to Madam Chair. Thank you very much.
Thank you for Professor Fazl for the great work. Your great work can resolve the problem of pollution in many countries. There's several questions from the participants. First question is the bacteria that can degrade halogenated pollutant, can they also use to bioremediation for heavy metals such as Ag, mercury, Ag, and etc. Thank you, Professor.
So basically, we never tried that on heavy metals because actually heavy metal is like something like they can attach to the cell surface. So we never tried that, how the heavy metals can remove using this bacteria. So basically, it is very nice to see the bacteria population sometimes involve other bacteria as well. So I'm not sure whether our bacteria can do that kind of job. So again, you talk about any special, because not only 2,2-dichloro or DL, so there are other toxic compounds as well involving aromatic compound. So again, we never test that. So if there is, you can always test that in various chemicals, but at this stage, it is very fundamental single proteins that we are interested in. So we wanted to characterize in terms of the acid amino, their structure and functions, and not more than that. So I appreciate that a lot of investigation can be done actually especially heavy metals is very demanding areas. Yeah, thank you very much for the question.
Can I ask you more question about related to the last statement? So this bacteria, is it genetically engineered or just indigenous species from the Lake Tas in Turkey, or is this a genetically engineering bacteria?
No. We don't involve any genetically bacteria here. It is all, we take the gene, also we do PCR direct from the genomic of the bacteria. We never do clone it, then we can always do something like selective mutagenesis, but we never to clone the gene at the moment. We just set this, okay, of course can be done at later stage.
Yeah, because the name of the bacteria is halophilia. So you just name the bacteria halophilia because they can degrade halogenated pollutants.
Standard Pseudomonas. Yes.
Oh, Pseudomonas halophilia. Okay. That's so interesting.
Yeah.
Next question.
For the next question, Professor, since some plastic wastewater also contain halogenated synthetic dyes, do you think these bacteria are also useful for bioremediation for plastic wastewater to apply in plastic industries in here in Indonesia?
You mean microplastic?
Plastic means.
I'm not very clear actually. Yeah, I'm not very clear, but you can always email me later on. We just embarking the microplastic expert research. I don't have a clear view about microplastic, but of course, sometimes microplastic involve more than one bacteria. So always aromatic involve more than one bacteria sometimes. Yeah.
Okay. Thank you very much, Professor.
Yeah.
So one question again, Professor. How did you know that the primer you have designed was good to identify the bacteria?
Sorry?
How did you know that the primer you have designed was good to identify the bacteria? Did you do the preliminary experimental first?
Oh, okay. The primer initially we took it from the published primer sequence. They use standard primers to fish out this dehydrogenase type gene, and then but then we obtain still not enough size that we expected from the gene. So therefore, using the partial sequence, we derive another primer. We do sequence comparisons, and then from there we managed to get another PCR products, and then we sequence it and get the full gene sequence. I mean that's the idea. I can't remember the actual primer design, but can check in the previous publications the primers that we use from the already published, and then the one that we design based on the partial sequence.
Okay, thank you, Professor. The question from Professor James Kertudat from SBT University. First question, do the bacteria produce any compatible solute to balance the osmotic pressure inside the bacteria?
Yeah, this is good question actually. We never tried that. If it can produce the principle, sometimes that's the idea. So how to survive? They must be able to produce the compatible solutes so that can balance. But if not, maybe they can utilize from the outside, can bring it into the cells. So there's so many parameters, but we never really go into it. Whether this kind of solutes some special genes can induce. I mean, can allow this solute so that it can balance the cells.
Okay, and the next question from Professor James. For your new project on the soda lake, what is the pH of the lake? If high, is the cytosolic pH similar or more neutral?
Yeah, the pH is 8.5, I think. It's above 7, yeah. Above 7, but definitely not 7, 8.5, things like that. To my recollection last time, yeah.
Okay, thank you. Because the limitation of the time, we will finish this first session. We thank you for Professor Fahrul for your presentation. We will go to the next presenter. Thank you, Professor Fahrul.
Thanks so much for the opportunity.
Applause to Professor Fahrul.
Thank you.
Ya, cepetan ya. Moderator ya.
Ladies and gentlemen, before we continue to the second speaker, we want to announce you to keep in your Zoom meeting because we want to take a picture for the second ISSMART 2020. Thank you.
Sudah. Dikasih hitungan 1, 2, 3 gitu.
All right, I will count. 1, 2, 3, go. All right, thank you.
Thank you very much, Professor Fahrul. Now, our second keynote speaker is Professor Dian Handayani, PhD from Department of Nutrition, Faculty of Medicine, Universitas Brawijaya. This is the curriculum vitae from Professor Dian. She's a professor of nutrition in Faculty of Medicine, Universitas Brawijaya. Also a lecturer in Department of Nutrition. From until now, she had a lot of publication about metabolic syndrome. Now we will begin the presentation for Professor Dian Handayani. Time is yours. Please welcome Professor Dian.
Aku sendiri yang mute. Sudah mute ya. Jadi saya
Okay, thank you. Good morning, everyone. Did my voice already can be here?
Yes, I can hear you, Bu Dian.
Okay, thank you very much. Okay, so good morning, the Honorable Vice Rector that already opened this session today, and also good morning to the first speaker. That's very interesting presentation, Professor Fahrul. And then firstly, I want to thank to the ISSMART committee that having me to this event this morning. I'm really appreciate with this invitation. And today I want to share my experience about Beta-Glucan. This is my research for I think around 10 hours after I finish my PhD. Did everyone can see my presentation?
Yes.
The screen is already can be seen?
Yes.
Okay. Okay, let me introduce myself. So my name is Dian Handayani. As Ibu Nia, as moderator introduce me. I come from nutrition department. So because I see that many participants from another country and also the speaker from another country, let me introduce where I come from. This is Malang City. This is the iconic place in Malang, and this is my university, the University of Brawijaya. And this is my faculty building. I came from medicine faculty, especially in nutrition department. So I want to talk about beta-glucan, but previously I want to give little bit introduction why I interest with this study. So basically my major research is about obesity, because obesity, as we know, this is still a global health problem. And then obesity also promote the metabolic syndrome. We can see in here that obesity is chronic disease. There are many factors can be grow by the obesity, like dyslipidemia, and then obesity will increasing the adipose tissue, and then affect to the insulin sensitivity and the promote metabolic syndrome. There are many studies established about this one. And our concern is about the obesity impact, because they are raising medical spending per capita. We know that obese people mostly have more medical costs than people without obesity. And whereas Indonesia in the midst of obesity epidemic, how Indonesia progress in obesity can be seen in this screen. So no place in the world can be immune from obesity, include Indonesia. Indonesia have around 24 to 32 prevalence of obesity. And this picture show how obesity in my country start in 2007, in 2018. There are no slow graphic in this picture. Both sex, women and men, have increasing obesity prevalency. And obesity is closely correlation with the type 2 diabetes. You can see in here the red dot and the green dot. This one, the survey in the different year, we can see in here in 2013 significantly increased the prevalence of obesity if compared with 2007, and the similar pattern in the diabetes prevalence. So that's why we concern about obesity, how to manage obesity to prevent the metabolic syndrome. So to prevent the obesity, there are many ways to prevent the obesity and metabolic syndrome. And I just concern with the community awareness in functional food that getting increased. We can see in here, there are many functional food that we use in the community to prevent obesity or metabolic syndrome. So the picture show this is oat, mushroom, and then some cereal and tea and whatever people just choose the good functional food. So little bit about functional food to make you understand about this one. People also know about nutraceutical, but nutraceutical is different with functional food, because nutraceutical is come from two words. This is nutrition and pharmaceutical, and the form in nutraceutical could be similar with the drugs like pill, extract, tablets, and etc. But for functional food, it's different. Food can be grouped as the functional food if retain food form and not form into pills or capsule. So for easy for us to understand deeper about this one, we can see in here. In basic food, the functional intensity is only for fulfill the basic needs, the physiological effect. and then little bit increase. If the food already enriched or enhanced food, they will have positive nutritional equilibrium. So the physiological effect will increase. And the increasing more higher than enriched food is called functional food that can reduce enhance function and contribute to restore health. And the nutraceutical in the top one. So I just, in my research, I use functional food for my intervention. And in the future, I also will make the nutraceutical to make a better the component of beta-glucan for cure or health. So just easy to remember, if we talk about functional food, it's still in food matrix, but if in nutraceutical, it will be non-food matrix. Okay, let's talk about the beta-glucan. Okay, wait. Buka pintunya. So the beta-glucan definition and characteristic. So why I interest with this one? Glucans are close polymer which have interchange linkage either alpha or beta link, and beta-glucan is a heterogeneous group of non-star polysaccharide consists of the glucose monomer links by beta-glucosidic bonds. Sorry. Okay, so the characteristic of beta-glucan can be seen in here. This beta-glucan is soluble fiber and fermentable. So that's why this look like suitable for the prevention of obesity. So soluble fiber, it means increased total transit time by the lion gastric emptying and also slow glucose absorption. And the characteristic for fermentable, it means glucan highly fermentable in the cecum and colon, that giving prebiotic effect, induce the maximum growth rate of bacteria and lactic acid production. Okay, let's go to the history about beta-glucan. So research in beta-glucan started in America and Europe in 1940, which is they see the immunomodulatory effect of zymosan. And then after deep intervention and research in 1970 and 1980, zymosan was examined in detail as a beta-glucan, as a component of primary. Little bit different with the American and Europe. In Asia, the history of beta-glucan is come from the Asia medicine. They consume the different medicinal mushroom like shiitake, maitake, and reishi, and then they realized in 1969 that beta-glucan isolation from shiitake mushroom content of lentinan. Okay, so this is source of beta-glucan. If we want to study about beta-glucan, there are many food that can be used as source of beta-glucan. They group as cereal like oats, barley, millets, and also come from mushroom like Pleuretus osteratus, or we can also call this one oyster mushroom, and then Pleuretus eringi mushroom, and then Lentino edudes mushroom, or popular with the name shiitake mushroom. And also another source of beta-glucan like microorganism like yeast, fungi, and bacteria, and also lichen, algae like Laminaria sp. So this is the source of beta-glucan that we can use as source for functional food. And structure of beta-glucan, of course, different in every group. Like bacteria have linear B13 backbone with no branches. For cereal group, have linear B13, B14 backbone with no branches. And seaweed have B13 backbone with B16 branches. In the mushroom, also a little bit different. They have B13 backbone with B16 branches, which is short in mushroom and fungi and long in yeast. So this is the structure of beta-glucan. The different food sources have different structure of beta-glucan. So just a little bit remember that research on beta-glucan, there are many. to see the functional food or nutraceutical that contain beta-glucan in immune response and also in metabolic syndrome. And I focus in metabolic syndrome. So this is the content of beta-glucan. We can see in here per 100 gram different food have different content of beta-glucan. The highest one is from wood ear mushroom that contain 41 gram per 100 gram. And then shiitake mushroom quite high also in 25 gram per 100 gram. And also the oat. There are many data from oat, 3 till 8 gram per 100 gram. Let's see the effect of functional food oat for combating metabolic syndrome. This study has been done with the oat to check the dyslipidemia effect and also the immune response. This is a study from Eleanor Beck. They see the effect of oat beta-glucan to increase postprandial cholecystokinin level. This is the gut hormone for anorexigenic hormone, how they can decrease insulin, and also how the subjective satiety after overweight subject consume oat beta-glucan. We can see the result in here. So, after consume the different doses beta-glucan, low doses, 1L, low, medium, then and high. The height one, the researcher used 2 different height one, but the content is the same, 5 gram of beta-glucan. We can see in here, after respondent eat the oat content of beta-glucan, the insulin area under curve getting decreased. This is good for preventing obesity because the insulinemia, hyperinsulinemia is closely related with the adiposity syndrome. And also the insulin, decreasing of insulin is also reported effective by increasing of CCK from this research. CCK is anorexigenic hormone, which is the increasing of CCK will increase the satiety for person that consume high fiber. So this research can be said good the oat for the prevention of obesity. Another study still used the functional food that contain beta-glucan. This study used beta-glucan in the 2 different doses, which the control very low or can ignore the beta-glucan content. This one, let me check meaning of PB. Okay, so the PB it's mean this is very high fiber, around 40% the flour replaced with the high fiber, and this one also high fiber, but in this group the beta-glucan enriched it means additional from outside, and this group only original have beta-glucan high. So how the effect? We can see in here. This is the delta. This is the delta of triglyceride-rich cholesterol. This is the delta. It's mean the changing. So after 1 hour, 2 hour consume these 2 group of high fiber food. The filled dot, it means the triangle one significantly different. It means the cholesterol decrease significantly a lot if compared between the food that very low fiber. And then another result, plasma cholecystokinin also show the same thing. In this study, they have like make conclusion. Let me see. The prolonged elevation of cholecystokinin may reflect the prolonged presence of a fat in the small intestine as a result of slower digestion and absorption of fat after eat high fiber meal. So high fiber meal will prevent the absorption of fat content of food, and then that's why the cholecystokinin have elevation prolonged if compared between the low fiber. And then another result also show the similar thing. It's very good effect of barley pasta that have beta glucan content. Similar with the oat, the beta glucan content in the barley can decrease significantly the plasma insulin. That's the functional food effect from oat and barley. And how effect in mushroom. This is based on my research when I'm doing my PhD. So this is shiitake mushroom. The shiitake mushroom, when I study in my university, I already analyzed the content of beta glucan almost 30%. It's quite high. And this food is very popular in the Chinese people to consume daily as part of their vegetable. So some study also established that the shiitake mushroom have effect in plasma lipid lowering and also increase the immune response. Interestingly, the shiitake with this structure also have another biological stuff that also useful for plasma lipid lowering and also antioxidant. This like chitin, ergotianin, erythadin, and other nutrients. But if we compare with the content of beta glucan is highest if compared with this biological stuff. So this is my study that compare between effect of oat and shiitake mushroom to prevent body weight gain in rats. So I use the in vivo model in rats fed with high fat diet. Let's check the result. So the result is interesting. When the rat give fat with oats and give fat with mushroom, if we compare the body weight gain is changing body weight after intervention, those two groups have significantly different. The body weight gain in the rat that food with mushroom significantly lower if compared with the oat group. Also, the similar thing in plasma triacylglycerol. In the plasma triacylglycerol, the mushroom group saw that this one decreased significantly if compared with the oat. Sorry. And the different color bar, it means different doses. And then if we check the total fat mass, including white adipose tissue, still significant difference between oat group and mushroom group, which is mushroom enrichment diet group had significant lower total fat mass and white adipose tissue if compared with the oat enrichment diet. So this really interesting. We just thinking, or maybe the erythadin and chitin also have the effect that can emphasize the effect of beta glucan. Continuing this study, I just checked the fecal fat of the rats fat experimental diets because we just thinking if the fat come to our food, it will make our body increase the body weight, or we have something can help our body to exclude the fat out from our body. And then this is the result. If we check the oat group, high dose oat and high dose mushroom, both of them have significantly increased the fat fecal if compared with the high fat dose only. So it means both of them, oat and mushroom, have similar way to exclude the fat from food to prevent the body weight gain. But why mushroom have very good effect if compared with the oat mushroom? After we analyze this, we can conclude that comparison between oat and mushroom, the mushroom diet is better than oat diet as part of high-fat diet because it reduces body weight gain, total fat mass, plasma triglyceride, and increased fat fecal excretion. And then the doses required for this effect was high dose. So the significant thing in mushroom is only in high dose. With lower doses, another one having minimal effects. So the potential reason why mushroom better than oats was possibly its chemical structure. We still analyze that one. And in addition, shiitake mushroom also contain another potential biological component, namely erythadine. Okay, then I continue to analyze how the shiitake mushroom can give the very good effect. So I just focus the effect of mushroom in body weight gain. They have negative correlation. It means the highest dose of the mushroom powder will give the bigger effect in body weight gain. That's happened also in white adipose tissue, plasma triglyceride, and also the body fat index. So the three possible mechanisms how shiitake mushroom improve obesity, we just thinking maybe because the increasing on fat elimination into phases is higher. Or we just thinking the decreasing of non-esterified fatty acid level, or erythadine from shiitake mushroom inhibit the release of triacylglycerol from the liver. We just emphasize in the number 3 one because in the fat elimination is already have data, and also the number 2. But we just curious why shiitake mushroom have very good effect to prevent the body weight gain. When we check in the liver, surprisingly, the fat weight in the liver is increased significantly if we compare with another group. So high dose mushroom give very high total fat in the liver. So we just continue. What happened with the total fat in the liver? So increase. So we do the pathology anatomy slicing liver, and we can see in here, surprisingly, the mushroom in high dose shiitake mushroom produce the lipid ballooning. There are many fat mass in the liver. So that's really not really good reason if we want to decrease our body weight, but we have problem in our liver. So we continue to analyze this one, and then the underlying mechanism of this shiitake mushroom has been published in my paper. So we want to assess the potential underlying mechanism of shiitake mushroom in lowering plasma triacylglycerol in red fat, high fat diet. So in brief, because if we talk too much, I'm sure the time is up. So I just make like a figure like this one to easy everyone understand with the underlying mechanism of shiitake mushroom. So in this study, we give high fat diet plus shiitake mushroom. We have data, the shiitake mushroom prevent the fat absorption that revealed by increasing of fat fecal that we have already the data. But we're still curious why, even though the fat already throughout in the feces, the body weight still decrease, and especially plasma triglyceride is still decreased. We know that the lipid, when come to our metabolism, they will be packaged as kilomicron, and then kilomicron remnant will be absorbed in the liver, and then the triacylglycerol will be put in the source of liver triacylglycerol, and also become a fatty acid, this one. So this is just normal mechanism. And then we know that erythadine from the shiitake, they have pathway to activate the CDP ethanolamine pathway. So this one, the CDP ethanolamine pathway, the CDP ethanolamine pathway in our liver, the function is to produce the phosphatidyl ethanolamine to increase. And then, because the erythadine, the phosphatidyl ethanolamine significantly increase, they will affect the ratio of phosphatidylcholine and phosphatidyletanolamine, which is this one is important to the health of the liver cell getting decreased because the PE increase, so the ratio will decrease. The decreasing of this ratio will affect the liver cell damage. And then the normal process, ApoB and source of triacylglycerol normally will be released as VLDL, will turn back infiltration to the liver cell damage and then produce the lipid ballooning. So that's why when we check the triacylglycerol in the liver, in the plasma from rat fat, high fat diet with the high dose shiitake, they don't have too much triacylglycerol. But when we open the liver and slice with pathology anatomy slash, we can see the lipid ballooning like this one. So this is the result from this study. So I have the data, the ratio of PC and PE decrease. Back a little bit to this one to make you remember, decrease. And this is the data significantly if we compare with the high fat dose only. And then the decreasing of PC and PE will affect to the liver cell damage. Or easy to us, I just try to make a figure like this one. This is the membrane or liver cell. The outer membrane content of PC, the inner membrane content of PE. When this ratio changing, the outer, the PC decrease and the PE increase, it will be happen the liver cell damage, and then the infiltration of whatever in the plasma will come to the liver, include the VLDL. So that's why the plasma lipid come to the liver, and then the lipid ballooning will produce. So how to prevent an EFLD then? We interest with the shiitake mushroom because good in prevention of obesity, but still have an EFLD. That's not good. So this is just what I'm thinking about that one. So we should make sure the choline diet is enough in our consumption if we eat the shiitake mushroom. Why? Because the choline diet will make the metabolism or phosphatidyletanolamine or CDP etanolamine pathway in normal way. So we can see in here, if choline diet is good, PC catabolism will produce in here. So our liver will have a number of PC, and then the acetylcholine metabolism will normal happen. So this one, erythadine, when we consume erythadine come from the shiitake mushroom, even though erythadine will affect on the CDP etanolamine pathway that increase the PE, it's no problem. So basically the erythadine will inhibit this mechanism. This is adenosyl homocysteine hydroxylase that promote adenosyl homocysteine to change into adenosyl methionine. If the erythadine established in our food, it will be inhibit. So that the PE will be inhibit to change into PC. And then if the choline diet is not enough, the PC not established in our liver, so it will happen in the red dash. This one, the red mark in here, if the choline deficiency and also if we eat erythadine too much. The PC will inhibit, the product of PC will inhibit because the CDP etanolamine pathway too much PE, and then the ApoB and triacylglycerol, if the normal way will assembly to produce VLDL, they will be inhibit, not produce VLDL, so that why our VLDL will decrease. So this is need further investigation. if we still interested with the shiitake mushroom to eat to prevent our body weight because the content of beta-glucan is very high, so we must consider to add choline diet in our food. That's our study that, of course, this is still not too long journey study. I think that's only 10 years, start from my beginning for PhD. So I still continue to this study. A little bit, I will want to make a conclusion about our study. So study in beta-glucan from oat combating metabolic syndrome, we can make a conclusion. Oat beta-glucan promising to decrease appetite such as CCK that release in response to beta-glucan at minimum dose. So it means 3.8 gram is sufficient enough to increase our satiety response. And then also insulin response relevant to develop of type 2 diabetes are significantly decreased at dose 3.8 of beta-glucan. This is based on research. And study from barley beta-glucan for combating metabolic syndrome is also promising, which is beta-glucan promote viscose polysaccharide in the diet. So they do slow the rate of carbohydrate and lipid digestion absorption. And the study also said that consumption of barley containing pasta, either naturally high or enriched, both of them lower bad cholesterol, support the hypothesis that viscose polysaccharide lower plasma cholesterol, in part by facilitating reverse cholesterol transport. And how about the comparison in shiitake and oat? We can make a conclusion like this one. The mushroom diet, yes, is better than oat diet as part in high-fat diet because it's already revealed that shiitake reduce body weight gain, total fat mass, plasma triglyceride, and increased fat fecal excretion. The doses required for this effect was high dose. In addition, should remember, mushroom, shiitake especially, contain another potential biological component, namely erythadine. And there are positive correlation between doses and liver triacylglycerol and liver ballooning histology. A negative association we found in mushroom doses because they affect on ratio PC and PE. So this study saw that mechanism how the high dose shiitake mushroom prevent obesity by increasing triacylglycerol accumulation in the liver rather than in adipose tissue. Whether or not this unwanted side effect can be prevented by addition choline to the diet need further investigation. So that's I want to share with you all my study about functional food that contain beta-glucan. I'm still committed to study a functional food, especially mushroom from beta-glucan. And now I study from oyster mushroom. I choose oyster mushroom because there are abundance in Indonesia. There are a lot in Indonesia, and Indonesian people just familiar with this food. But I also want to check the gut macrobiota, how it works, because we know that there are good and bad microbiota, and that's affect on our tummy health. So this is our friend and our colleagues, our teacher that already support me and support us to study with this one. Thank you very much for this your attention, and this is my references. Any discussion I welcome, and then I send back this time to moderator. Thank you very much.
Thank you very much, Professor Dian. Very interesting research, since we know that obesity and metabolic syndrome is the major comorbidity for COVID-19 in this pandemic. I will deliver a question from the participants. First question is, when we conducted a diet management with no intake carbohydrate at all, only vegetable and fruit, what is the impact to our body in related to health?
Sorry, Bunia, I just focus bit close my presentation, not listen well your question. Could you repeat again? I'm sorry.
Okay, thank you. I will repeat the question. If we conducted a diet management with no intake carbohydrate at all, only vegetable and fruit, what is the impact to our body in related to health?
Okay. Okay, thank you, Bunia. Very interesting question. Little bit different with my study, yeah? So you asked about if we remove the carbohydrate at all from our diet. Because I'm a dietitian, so in my perspective, we not suggested to remove the carbohydrate from our diet because our body still need carbohydrate for the energy source in our brain. At least 130 grams per day should be fulfilled in our diet. But we must understand that carbohydrate not only come from the staple food like rice, like potato, like what people say, rice, potato, and etc. that high in carbohydrate, bread or something. But vegetable also have the carbohydrate. So if you eat a lot of vegetable, you also have carbohydrate, but of course not too much. So the trend diet at the moment, like keto, this is not my expertise, but I'm not suggest take the keto diet without medical doctor pengawasan itu apa? Pengawasan, not medical doctor supervision, because without monitoring from medical doctor, we just wonder if there are too much ketone bodies increase in our body, it will be dangerous for our body. So as long as your ketone body is okay, ketone diet is okay. But if ketone bodies high too much, it will dangerous for our body. So be careful if running the keto diet. We should make sure to check your ketone body, because basically the keto diet is for the people with the have problem to metabolic in carbohydrate, like people with what we call apa Bunia? Sorry, I'm forget epilepsy. Okay, I think that's my answer.
Okay, we go to the next question from Professor James Keto Diet, SUT Thailand. First question, Professor Dian, is increased consumption of dietary beta-glucans likely contributing to increased non-alcoholic fatty liver disease in the world?
Okay, Prof. James have two questions, Ibu Nia. Should I answer one by one or just because I can read this in my screen? Is it okay to answer directly, Bu Nia?
Okay, two questions.
Okay, thank you for Prof. James for your question. This is no. Dietary beta-glucan is not likely contribute to increase non-alcoholic fatty liver. No, because in my study, why they increase fatty liver? Because I use shiitake mushroom that contain erythadine. If the product not contain erythadine, of course, this is no produce the non-alcoholic fatty liver. This is my understanding, Prof. James. So what make happen problem in my study because the erythadine content from shiitake mushroom. I think that's my answer for the first question. And then the second one, do fermentation product of the colonic microorganisms such butyric acid affect? Yes, yes, Prof, this is correct. So the fermentation colonic microorganism will produce saturated chain fatty acid. Which one exactly, I'm not really remember because I'm still studying about that one. The SCFA will affect the FFRR3. This is the receptor for the satiety. So if the fermentation have increased the FFRR3, they will affect the increasing of satiety. So this is very good to have the fermentation product colonic microorganism to our tummy, to our gut, like that, because it will affect on the satiety hormone, produce anorexigenic of satiety hormone, like PYY, something like that.
Thank you. Another question
From Bu Anna, ya.
Bu Anna and Mrs. Cleonara.
Okay. I answer directly, Ibu Nia?
Yes.
Okay. Thank you, Bu Nia. So, a question from Bu Anna. Do we need to consume choline together? Yes, Bu Anna. Based on my study, it suggests like that one. But, of course, this happen in high dose of mushroom. Basically, people not consume mushroom too much in our country. But it will be good if we consume together with choline. But sorry, I cannot explain clearly how many choline. Choline basically from vitamin B, Bu Anna. So I cannot describe clearly how much the dose, because it needs further investigation. But yes, if you consume good choline from the vitamin B, it will decrease the effect of erythadine that can produce the fatty liver. Okay, and then thank you. Oh, Bu Cleo, ya, Bu Cleo Nara, how many percent fat each from feces? Bu Cleo, thank you for your question. Maybe we just calculate carefully in the human, but in our studies show that the feces 19%. Sorry, let's back to our data. Wait a second. 19% fat excretion per 24 hour from rat, Bu Cleo. Yes, so this one 19% from total fat, from total feces in 24 hour for rat that consume high fat diet. I think that's all, Bu Nia, the question.
Okay, thank you, Professor Dian.
Yes.
This is the last question for this session. Thank you very much for your explanation and your presentation, Professor Dian.
My
please
pleasure.
applause to Professor Dian Handayani, PhD. That's all our first plenary session. Thank you for Professor Fahrul and Professor Dian and for all the participants. Now we are going to the Master of Ceremony to handle the next agenda.
All right, thank you, ladies and gentlemen. Now we are continuing to the next agenda, that is oral session. And we inform you to please enter the each link as shown in the screen based on the screen. Ladies and gentlemen, and we have 15 minutes time for break to enter for the each link as shown in the screen. Thank you.
you.